The planet Earth, as we know, is home to millions of different species, and the variation among the species can be defined as the term biodiversity. From the small insects you see every day, to elephants and lions in the safari, and to the aquatic creatures in the ocean, we're surrounded by biodiversity. We're all known to the fact that species evolve over time in different environments. However, even the same species can evolve differently under different environmental conditions, also known as phenotypic plasticity. Phenotypic plasticity can be defined as the characteristic of individual genotypes to give rise to different phenotypes when faced with different environmental conditions. Likewise, there are organisms that portray different phenotypes in response to selections, predators, and other resources. A knowledge gap that exists is a complete understanding of the genetic architecture that underlies the phenotypic plasticity within diverse environmental conditions. There is a lack of observed studies that exist which relates to the measure of genetic basis of plasticity in complex environments. Another knowledge gap in the field of phenotypic plasticity is that the majority of studies focus on the effects of a single environmental factor on the phenotype of a certain organism. They don't take into account the fact that organisms are a part of complex ecological system and they're constantly affected by the interaction of various environmental factors involved in their growth, development, and life histories. Furthermore, a knowledge gap that remains to be studied is whether plastic traits are always beneficial when they're expressed in the appropriate environment, if they're a product of divergent selection, or if they vary among species as stated by the hypothesis of adaptive plasticity. Similar to various living beings on this planet, wood frogs are commonly found in eastern and north-central United States and all over Canada. The tadpoles of wood frogs are commonly eaten by insects, and correspondingly they have developed several anti-predator mechanisms to protect themselves.
Larvae of amphibians have shown to undergo phenotypic plasticity throughout their life in terms of diet, behavior, and morphology when faced with environmental changes involving factors such as predator and food. These knowledge gaps have been addressed using the range of traits such as tail fins and body size in respect to the predator-induced environments. These scientists rely upon using species with adaptive morphological characters in response to the particular environmental change to obtain reasonable results. As an evolutionary biologist, we are raised with questions such as, does phenotypic plasticity really exist in amphibians such as wood frogs? And if it does, to what extent? To answer these questions, Busker and Relia conducted a curiosity-driven experiment in wood frogs, also known as Rana sylvatica. The purpose of their experiment was to determine if phenotypic plasticity involves an adaptation to different predator environments. Their study focused on the hypothesis that natural selection currently acts on plastic traits and maintains plasticity by favoring different phenotypes in different environments. In particular, they wanted to do an experiment to underline if the plasticity in shapes of the tadpoles is maintained by divergent selection in the presence and absence of predators. The scientists collected eggs of Rana sylvatica from 8 different populations in April 1996 in southeastern Michigan. As wood frogs are known to produce a mass of eggs, it was very important that the eggs be placed separately until they were hatched and ready for further investigation. 3 groups of full sieves were taken from each population. The study contained different phases. Firstly, to determine the change in the morphological plasticity with the absence of predators, and then to examine if the predators chose the tadpoles in respect to their size and shape. Outdoor artificial ponds with naturally found nutrition such as leaf litter and other food resources from nearby habitats were accumulated to provide an initial source for the experiment. Tanks were protected from unwanted predators using fiberglass. The study included 24 replicates of each group of 110 full sieves, where there was either the presence or the absence of predators, respectively. After 18 days, 20 tadpoles from each tank were removed for further analysis. Those 20 were then divided into groups of 2 and were placed into a plastic tub. The tub had a 14:10 ratio of light-dark and contained a coil of plastic rope to provide purchase for their predators. A single annex starved for 2 days was then placed in the respective tubs and removed the following day, and the surviving tadpoles were preserved in 10% formalin. The morphological characters were then differentiated, mostly to determine whether the predators killed tadpoles based on their size and shape. The morphology of tadpoles were measured using a dissecting scope and camera connected to a computer equipped with image analysis software. The length and depth of the body and the maximum depth of the tail fin and tail muscle depth were then digitized. The ones that had no predators were used as a reference, and the ones in trials were used to estimate the resulting phenotype.
According to the results, in 18 days, major plasticity was observed in the body and tail shape of Rhina sylvatica. It was revealed that the tadpoles showed high differences mostly in their body length and tail fin depth based on the multivariate analysis of all 7 traits monitored. Additionally, the tadpoles that were exposed to predators had short bodies and long deep tails in comparison to the ones that were not. However, although various differences were seen in the tank with predators, no consistent relationship was gathered between the phenotype and performance of tadpoles in the tank with no predators. It has been found that despite some inconsistencies related to the interaction of different factors, including predatory range and species diversities, plastic traits vary among species as stated by the adaptive plasticity hypothesis. The study comprises various figures that show relationship between the variables. The figures are standalone as the caption provides enough information for each given figure. One of the figures is shown here. The shown example here is figure 5 from this study. In this table, the x-axis is labeled as plasticity coefficient and y-axis is labeled as selection coefficient. The predator presence is mentioned as well. The coefficients are a measure of the strength of plasticity. The traits with positive values were noted as larger in no predator environment. It is clear that there is a significant relationship between the plasticity and selection for 7 measures of Rhina sylvatica tadpoles. The use of illustration in this study helps us gather more information on the topic and build upon our theoretical knowledge obtained so far.
The main findings of the study were that tadpoles from the 2 different environments had different morphological features and indeed showed plasticity in body and tail shape. The hypothesis of plasticity represents an adaptation to environmental variability was supported, with a close alignment of the traits observed in tadpoles in presence of the predators. However, the inconsistency in the results with the predator-free tadpoles contradicts with the hypothesis. If plasticity was adaptive, then different traits should show high relative fitness in different environments. However, the predator-free tadpoles did not have consistent traits. Various other factors such as the predator range and the species diversity and so on need to be taken into account for further results. Some species adapt poorly in some environments and can generate different responses, whereas some species adapt in outspread environments. This evidence suggests that same species in 2 different populations can respond differently to similar cues under trackable environmental changes, which remains to be identified in the study. This knowledge gap can be used to generalize why tadpoles in our study might have shown different characteristics, because the experimenter created an environment for them where certain factors were not highlighted. It could have resulted in distinct responses. This way, by using an experimental study related to wood frog Rhina sylvatica, we were able to expand our understanding on the topic of phenotypic plasticity. Through the study, we were able to gain insights on how different organisms portray different phenotypes.
The results achieved from this study has furthered our knowledge significantly. The authors provided an exceptional analysis of the results, which have been used in other publications as well. For example, in 2011, another similar study was performed in Rhina sylvatica by Ryan Kulsbeck and Sean Costa, where they used the knowledge provided from the article to present similar study on the predator-mediated solution and development in Rhina sylvatica. Their study highlights how the survival rate of Rhina sylvatica differs in metamorphosis stages when they face predation by dragonflies. If this were the case, then our study remains to acknowledge the different stages of tadpoles and the correlation between survival and plasticity. From all the experiments, study reviews, and publications, we have learned that plasticity is plausible and can generate variation in different organisms. Furthermore, from an evolutionary perspective, today's environment is changing every day and species are slowly evolving. Now, are tadpoles the only one that adjust to changing environmental cues? That's another research still waiting to be explored.